Thank you very much for joining us in this panel. And I really enjoyed Patrick's talk and Anthony's talk. So there are definitely lots of interesting grading opportunities. I was basically updating my slides while they have been presenting. I was thinking, okay, how can I basically add other things or update the things that we are going to discuss? So the question I'm going to address today is, how can we use MRI or fMRI, what we have been discussing about all those interesting biomarkers in our daily clinical practice? Okay? How many of you in this hall are actually seeing patients? Okay, almost everybody in the hall, you are seeing patients, and you do clinical kind of practice. How frequently do you use MRI in your clinical practice in the field of addiction? Okay, so almost zero, right? So we do not use MRI or what we are going to discuss about fMRI in daily clinical practice in addiction medicine. And the question is why and how it could be changed. So that is the question I'm going to address and see how we can collaborate on that together. No conflict of interest. As Anthony mentioned, I have my slides on the QR code. We are also recording the session and we put them on YouTube. So you have access to the material, so you can just scan the QR code and you will have access to the slides. I will provide the QR code at the end as well. No conflict of interest. As Falk mentioned, I'm based in University of Minnesota, and I'm not sure if you heard that University of Minnesota has one of the largest brain imaging centers in the world, which is the site that the first fMRI studies that you see, Ogawa, Oakville, those people, they are basically based in University of Minnesota, and they have been able to do lots of fantastic jobs in brain imaging. And we have the world's largest MRI scanner, 10.5 kind of Tesla scanner. It's a huge scanner, which is located in University of Minnesota. And University of Minnesota also invested significantly in the field of addiction and addiction research. Mark Thomas, who is here, the director of medical discovery team on addiction, fantastic group of people who are working in this field, and an army of students and PhD students and residents who have been collaborating on the research I'm presenting today. Without their support and contribution, it was impossible to discuss about the things that we are discussing about today. But going to the question of MRI and fMRI and how we can use them in the clinical practice. Starting with really basic question, what is MRI? Okay, so we know what is MRI, kind of giving us an idea about what is the shape of the brain and how things are happening in the brain. And then we know what is fMRI. And in fMRI, we have rest fMRI and task fMRI. In the morning session, the discussion were mainly around the rest fMRI, when people are supposed to not do any specific thing inside the scanner. And what we are discussing today is mainly task fMRI. What we do is we expose people to drug-related cues, and then we also expose them to neutral cues, and we make the contrast between these two. And this is the map that you see in terms of these are the activations that you see as a result of that specific task-based data that we collected. And also, we can do connectivity analysis, or we can show you just the activation. So these are the two types of data analysis that we can have. We can just show you how things are activated or how things are connected. So these are the major types of fMRI that we potentially can use in the clinical practice. And this is the figure that you are seeing is fMRI with task, and this is the activation of the brain, not the connection. I can show you the connection as well. But how frequently fMRI is being used in clinical trials in the real world? If you look into clinicaltrials.gov and look for studies that they use fMRI or brain imaging in general, neuroimaging in general, as an outcome measure to test an intervention specifically in the field of addiction. Any estimate how many trials that we have that they have fMRI or neuroimaging as one of their outcome measures? 3. A lot, like 10, 20, 2,000? Number-wise? Of 100. 409 trials we found in the end of 2022, trials that they have been using neuroimaging as an outcome measure, okay? And you can see here in alcohol, nicotine, cocaine, and the number is increasing over the time, as you can see here in terms of the trials that are already registered in clinicaltrial.gov. And when you go to the details of those neuroimaging measures, as you can see here, the modality, the most frequent modality is fMRI. People are using EEG, structural MRI, PET, SPECT, other modality. The most frequent modality is fMRI. And the most frequent type of fMRI is task-based fMRI. And the most frequent type of task-based fMRI is drug cue reactivity. So drug cue reactivity is the most frequent version of fMRI that is being used in the actual assessment of outcome measures in clinical trials. So this is the most frequent, but there are other ways of activating brain circuits that are related to addiction. There are other tasks that are being used. We have done a recent systematic review on all studies that they did fMRI drug cue reactivity with a large group of colleagues, 2 brilliant postdocs in my lab, Mehran and Arshia, and also many colleagues, including Falk and kind of Patrick and Antonio and many other colleagues, Sabine and kind of all those other colleagues that we have here, and all those people that have been contributing to that sort of large effort. And we have been trying to see, okay, what is the status of the field? What is happening? How fMRI drug cue reactivity, after 25 years of using fMRI drug cue reactivity, do we have any clinical application for that? And we discussed there that so far, there is no single FDA approved application for fMRI drug cue reactivity or any type of fMRI in addiction medicine. Okay, so that is the current status of the field. And we need to face the reality. And the question is why and how this might be kind of different in the future. To have a better idea about the current status and what should we do in the future, just let's think about how neuroimaging as a biomarker could be used in the clinical practice of addiction medicine, okay? So this is the process of a healthy person starting to use substances, developing substance use disorder, going to a specific intervention, and making a specific treatment outcome, or many of our patients, they are actually not receiving a specific intervention, but over the time they recover or they relapse or the sort of natural course of substance use disorder that they experience, okay? So this is an overview of a disease that we are facing and we are working on. We call it substance use disorder, right? Do you agree? That is the overall view. And which part of this process brain imaging could be helpful in your clinical practice? Just think about, you are facing people in different levels of this disease, right? So how neuroimaging or fMRI, or what Patrick was presenting today in terms of the fMRI, the activation of the brain response to drug cues or the activation related to craving, how that is going to be useful in your clinical practice? How do you want to use that? If I give you the map, how you are going to use the map in your daily clinical practice? Okay, just to start thinking about that. There are specific guidelines. One of the most famous guidelines is BEST guideline for biomarker definition. And when they are discussing about the biomarker, they say, when you say neuroimaging as a biomarker, doesn't have any meaning. Do not use neuroimaging as a biomarker. Biomarker by its own term doesn't have any meaning. You need to define what type of biomarker you are talking about. And you need to say, okay, I can use neuroimaging as a susceptibility biomarker. What does it mean, susceptibility biomarker? It means if I do fMRI in somebody who has not started to use substances, or they are occasionally smoke cannabis, or they are occasionally drink alcohol, can we predict who is going to be more susceptible to develop cannabis use disorder? Who is going to be more susceptible to develop alcohol use disorder? Who is going to be more susceptible to smoke cannabis and then develop psychosis and schizophrenia? Could it be a clinically meaningful way of using fMRI? We can discuss about that. But what we call it is we call it susceptibility biomarker, okay? We can use it as a predictive biomarker. In terms of, when we are discussing about predictive, we are talking about a specific type of intervention. We are going to give people naltrexone or acamprosate or antabuse, disulfiram, different medications that we have for alcohol. Any chance that before starting these medications, based on the fMRI, we say, okay, because this patient has activation in ventral striatum, this patient is going to be responding better to naltrexone, not acamprosate, not disulfiram, we need to be focused on naltrexone and just optimize the dose of naltrexone. Is there any chance that we can use that in terms of deciding which treatment is going to be more helpful? That could be another way of doing that. Or how can we measure, we are already providing a patient with naltrexone, and the patient is saying, okay, I feel better, I do not have that much of craving, I have not been didn't have heavy drinking recently. You want to measure, am I very successful in the kind of addressing all the aspects of craving, or the treatment response is not complete, and I need to add another thing on the top of that. So you want to check the response to the intervention that you have and see, okay, have you addressed all the aspects of craving based on what you see in the brain, or you need to add other interventions to boost up the intervention that you. So thinking about using fMRI as a response biomarker to measure whether you already have enough response to treatment. So these are the different ways that you can use fMRI as a biomarker, and we can think about them and see how much evidence that we have to use fMRI in different levels of biomarker. Let's go to the next level. Any estimate in terms of number of fMRI drug reactivity that we have? We have been discussing about that just recently. So we have over 400 papers in the field so far, okay? When we are looking into these 400 papers, how these 400 studies use fMRI, which type of biomarker they try to discover using fMRI. Okay? If you want to go and see whether they use fMRI as a response biomarker, as a predictive biomarker, as a susceptibility biomarker, as a prognostic biomarker, as a diagnostic biomarker, what type of biomarkers they tested in their studies. So these are the studies, as you can see here, the number is increasing. We have nicotine, alcohol, cocaine, opioid, all those studies that are happening in the field. So if you look into this study from this framework and look into what you have in these studies, you can see that diagnostic, response, severity, prognostic, predictive, monitoring, susceptibility biomarkers, these studies are providing evidence for all those different types of biomarkers across different substances, okay? And even if you look into whether the results that they provided is significant or non-significant, many of them, they provided significant results. So as you can imagine, most of the studies use fMRI as a sort of diagnostic in terms of whether it can differentiate between substance users and non-substance users, or heavy alcohol drinkers and social alcohol drinkers. They sort of just differentiating between case and control, just trying to use that as a diagnostic measure. But there are many response, many severity biomarker, many prognostic. So there are multiple studies that they try to address and use fMRI drug therapy specifically for these sort of biomarkers. But there is a problem. What is the problem? When you go and see the details of these studies, I mean, even many of these studies, they have not clearly reported what they have done. There is not enough transparency. People have different labs, they have used different tasks, different kind of ways of doing fMRI. Single task that we are talking about, fMRI drug, there is a huge level of variability across different labs and different fMRI tasks that people use. So we try to put people together and try to develop a checklist together to agree to at least report exactly what we are doing in different labs to make sure that at least we know what is happening. But honestly, there is no sort of level of harm. So we are trying now to see how labs can collaborate together and kind of harmonize the ways that they are doing this. So going to these biomarkers that we have, 400 biomarkers I was discussing and 411 studies. When you go to the details of them, as I told you, when you want to specify the details of those biomarkers, there are lots of variations. Many studies, they use visual cues, but there are other cues that people use. Many studies, they use block design tasks, but there are lots of other different tasks. So there is a huge level of variation across different studies. And then when you go to the context of the use, which substance they are addressing, what type of biomarker that they are addressing, there are diverse set of different things. So the level of evidence for one single specification, one single context of use is just very limited. So when you want to define a biomarker, one important point I hope that you can have from this discussion, when you need to clearly define the specification of the biomarker and COU of the biomarker, context of use of the biomarker. And when you put these together, the studies that they are replicating each other and accumulating evidence, they are limited because they are distributed across the field. And we need to get them together, and what we are doing in Enigma, we are trying to do mega-analysis, adding them together and see how we can put these pieces of evidence. Because at the end of the day, what we need to do, we need to go to regulatory agencies and say, we have enough evidence that this measure can be used for this specific application in the clinical practice. So what FDA asks us, if you want to go to FDA and say, okay, fMRI drug therapy could be used for this specific application in clinical practice, they want you, this is a pipeline that they have. They want you to define the biomarker clearly, define the context of use clearly, and then go to what they call, they have a specific biomarker qualification pathway. And it takes time. People need to work together, they need to apply, they need to ask for specific support from. So you need to have a team of people working together and make that happen. Without large level of collaboration, it does not happen unless, there could be an industry. Most of the other biomarkers that we already have in the field, they have been supported by industry. So there was a support from industry that made those things happen. When we want to do that in the academic level, it gets a little bit more complex because we do not usually collaborate very much. So we need to bring all our forces together. It's a discussion that we already have with Patrick and Falk, to see how we can bring our forces together. Some like even kind of transatlantic collaboration with what we have in the US, what is happening in Europe, in UK, how we can bring our forces together to be able to apply for receiving that qualification. And even thinking about, okay, if we want to get focused to one or two of those specific biomarkers, not to just distribute our resources, what could be the best of the best from these biomarkers that we have? Which one is going to be more meaningful in the clinical practice? Okay? So one last piece of my discussion. Right now, there is a new development in the field of neuroscience and psychiatry, and we have new brain stimulation technologies that we can use whatever brain mapping measures that we have to actually do the interventions on those circuits, okay? So these brain stimulation technologies, as an example, electrical stimulation, magnetic stimulation, the number of studies are exploding, okay? And now people are realizing in different fields that we can use the measurement, the activation, the fMRI that we had from previous studies to use them as targeting biomarker. And targeting biomarker is a new definition in the field. Now, we use all the information that we have to actually target those areas and put the electricity there and modulate them and change the scenario. The first fMRI-based biomarker treatment received FDA approval in 2022. Anybody knows what is that treatment? What is that specific protocol that received FDA approval? fMRI-based, biomarker-based treatment in psychiatry that received FDA approval in 2022. Same protocol. So the same protocol powered by a company, Magnus Medical, is the first time that fMRI is a part of a treatment in psychiatry, okay? And they do individualized fMRI measurement of connectivity between DLPFC and subgenual cingulate cortex and target that for addiction, for depression treatment. And they showed really good results. I mean, it's an ongoing field that people are working on. But what would be the targeting biomarker in the field of addiction? And that is another really interesting discussion, that what are the networks that should be targeted, and the activation that we have, how that could be targeted. Or even going one level higher, what we are doing in my lab, trying to see how we can even individually find the right target. So when we are doing fMRI drug therapy, measuring the connectivity, these are 50 methamphetamine users that we are finding their best target to stimulate their prefrontal cortex, the frontopolar cortex, to have access to their amygdala. And we showed that there is a large variation in terms of the space for each individual to find an individualized target for people to have access to that part of the brain. And we showed that these individualized connectivities would be related to the level of craving that people are experiencing. And we are trying to basically use that individualized targeting biomarker using fMRI and we optimize them based on location, based on the way of the stimulation, the orientation of the stimulation, and also the dose of stimulation. And now we use robotic arm and robots to be able to find exactly the best target and do the stimulation exactly on the right target and individualized level. And also we optimize the dose. So now we are adding another level to the best guideline, and we call it target biomarker. And we are trying to even define what do we mean by target biomarker. But I think that I gave you an understanding in terms of potentially one of the areas that I see sort of low-hanging fruits or more hopes even in the future in terms of being able to add something that is going to be actually used in the clinical practice is the idea of targeting biomarker. Tomorrow, 9 a.m. in the morning, we will have a specific panel discussing about potential brain circuits that could be targeted for addiction therapeutics. If you are interested, you can definitely join us there. We had webinars discussing about that. This webinar, if you scan the QR code, is already available on YouTube. We discussed about personalized, non-invasive brain stimulation targets for addiction medicine. That specific discussion is already on YouTube. You can have access to. We will have a workshop using MRI to optimize brain stimulation in Brisbane, in Australia, in a month. If you are in Australia, you're welcome to join us in Brisbane. So we will have discussions there about addiction therapeutics and also how to use MRI to optimize brain stimulation. Different discussions. There are lots of technical details, but it's an interesting and exciting area for collaboration. As my colleagues mentioned, Addiction Cure Reactivity Initiative Network, we have the website on University of Minnesota kind of a platform. You can have access. You are absolutely welcome to join us. We are looking forward to have further collaboration and do things together in the future. ISAM, the Neuroscience Interest Group in ISAM, we developed a roadmap with Antonio, my co-chair in Neuroscience Interest Group, and we are very hopeful that we have the entire community of addiction physicians to be involved in this sort of development. We need to work together if we want to have the actual kind of development in the field. And I'm really happy that within ISAM, we already have experts from 100 countries that are working together within this society. And I'm hopeful that we can also work together to be able to make kind of new advances in the field and encourage and engage residents, medical students in the process. I was in Lithuania. Darius invited me just a few weeks ago in Lithuania. We had a workshop with their residents and psychiatrists. It was a fantastic experience of 2 days of brain stimulation workshop discussing about things that could be done. I was in Qatar just kind of 2 months ago. Rama invited me, and we are trying to develop a network of people who are interested to work together and do things together. take-home notes. My slides QR code is available. Today we discuss about what is fMRI. We discuss about fMRI in the clinical practice. We discuss as fMRI as a biomarker alone is meaningless. We need to define two items. We should specify and define the context of use. We need to say, okay, this specific task-based fMRI, Dirac-Curie activity, activation in this area, you need to specify that and then define the context of use. Then we can say it is this type of biomarker, okay? And then we discuss about this biomarker specification, we discuss about context of use, we discuss about FDA biomarker qualification pathway, and we discuss about fMRI as a targeting biomarker, as a new development in the field. And as Patrick beautifully mentioned, reliability is very important. So that is another level that we are working in terms of making sure the target that we have or the measures that we have are going to be valid, reliable, and how we can then make them individualized. And there are hopefully potentials for individualized interventions and potentials for networking. Thank you very much for your attention.